So, my 13-year-old son has a diagnosis of ADHD, severe anxiety, sensory processing disorder, and autism. In my experience, autism is rarely singular, and as every parent and carer knows, you meet one person with autism and you've only met and had an experience with one person with autism. So let me tell you a little bit about our Quinn. He is violent and he's incredibly loving. He's a comedian, he's political, he's fiercely loyal, family orientated, creative, can never be touched unless he initiates it, is good with English, repetitive, he is occasionally suicidal and often delightfully clever. He is simple, he is complicated, he smells because he's a 13-year-old boy. He eats a lot of sugar and anything else he can get his hands on. He swears a lot and he is a human being. He finds it very difficult to self-regulate. He does not sleep when we want him to. He spends most of his life on the PlayStation. It's hard to get him out of the house. He seems to have, in some ways, given up on people. He cannot eat or binge eats. He cannot take his tablets, get up from his bed, brush his teeth or engage in general hygiene without hands-on prompting. He says very embarrassing things in public. One in particular was when he asked what the letter was for my bra size on a bra. He said, "What's the letter for?" And I said, "The letter is for just to let you know what size the breast cup is." And we were walking down the street and a man inappropriately looked at my chest and Quinn leant over and said, "It's an E cup," in case you were wondering. And the man said, "Sorry," and I said, nothing. And we quickly moved along. But that's a great example of how Quinn doesn't quite know what's appropriate and what isn't. He's a very lonely child and he has not one friend besides those he's never physically met but has developed on the PlayStation, and he goes through those pretty quickly. And his behaviour over time has alienated many friends and family. He's a 13-year-old boy. Society doesn't seem to want him. He's been isolated from his schooling environment and forced into a special school, which changed his life for the better, but also exacerbated his social isolation. We often say that he would be just fine if it wasn't for everybody else. Mainstream school destabilised his sense of self, and one that we worked so hard to engender and work so hard to engender now. I've worked at the Florey in various forms for nearly 12 years, and in this time I've gotten to know Dr. Emma Burrows pretty well. When I first met her, I was in a steep learning curve about difference and different kind of brains, and I was still trying to make my son at that stage fit his very round self into a very square peg. I was drawn to Emma because of her passion for exploring human behaviour and her ability to look at every scenario with humour, kindness, and a mind wide open. I'm very excited by her research because I know what drives her is not a cure for something that makes my child and all autistic people so very special, but her passion for understanding why. Quinn wasn't able to come today. He was far too anxious and he's not often able to leave our house, but he wrote something for tonight that I'll read on his behalf. It's a bit funny, but I'm reading it exactly the way that he's given it to me as I was told to do. My name is Quinn and I suffer from autism. Psych! Autism is actually a gift, and it's a gift that only some people have, but a gift nonetheless. I think autism research is good, but I don't think its purpose is for a cure. It doesn't need a cure because it's not bad. Research should be for helping the community understand people with autism. If somebody looks different, acts different, speaks differently, they should be understood and not looked at like they're weird or wrong. Autism research, like the research Emma's doing, is important because it means people with autism don't have to change to fit in and be educated and play sport and be employed. It means the world around us will understand us, so it will allow us spaces where we fit and can be happy. I'm autistic, but I don't feel different. It's only the people around me who make me feel different, like I know I'm out of place. But if I'm out of place here, then where is my place? I wish people would understand when I don't get things, and I wish they knew that I can't ask for help or explanations, that I don't always know when I don't know. I hope that the advances in research will make a space for me and people like me to belong. I don't need someone to explain to me who I am, but I know if I'm going to have a better life, and others less lucky than me who self-medicate and struggle to like themselves are going to have a better life, then we need to be accepted for ourselves, not to have separate places where we are hidden away. Thank you for listening to I Guess My Mum talk for me. And if you can, and you have lots of money, fund Emma Burrows so she can do it quicker. Thank you. And so now I'll ask Emma to come up and give us her excellent presentation. Thanks, Emma.
I just want to begin by thanking Carmen and also Quinn. I was really touched by your words. They're brave and courageous and really generous. Thank you. Okay, so some of you may be here because you have autism. Some of you may be here because you know someone who has autism. Or some of you may just be curious. I thought I'd give you just a really quick explanation for why I'm here. Clearly, I'm an autism researcher, but I wasn't always one. And I owe a lot of my journey to my parents, who are educators and have dedicated their whole entire lives to working with people who have additional needs or need special help. And through their connections, I was really lucky as a 16-year-old to begin my working career as someone who was very important in integrating two boys into schooling, and this is something that my family are so passionate about, and I've definitely caught that. These two boys were really different and really fascinating, and they taught me a lot. Sam was very curious about everything. So what we did is we harnessed his curiosity to help him integrate into high school, and we worked together, Sam and I, on these amazingly in-depth investigative journeys to debunk mythical creatures. And when I met him initially, he had already debunked the Loch Ness Monster, and our journey was all about the New Jersey Devil, which is a very unusual creature. It's a bit like a kangaroo. It's a mismatch of many different things. And we actually went through a lot of sightings and looked at these sightings and reports analytically, and together we forged an interesting friendship, and we learnt a lot about the New Jersey Devil, and I don't think we actually ended up determining whether it was a real thing or not. But he was a very fascinating boy to me because of his interests, but also because we couldn't work in rooms with air conditioning, and we couldn't do any maths that ended in 6, and up until our interaction, he was not able to work with anyone that had a name starting with E. So I think we definitely worked well together, but there were some really interesting things about him that made it very difficult for him to integrate into a classroom where most of the people in that classroom were neurotypical. The second boy I worked with, I didn't have as much to do with, but I did learn still quite a lot about him. And Brian was very, very interested in the word fish. It brought him extreme joy. He was terrified of Saturn, and he also really, really liked to vacuum. So in order to get him into a class, he was often given the carrot at the end of the stick was you get to vacuum the classroom at the end. So these two boys both had a diagnosis of autism, but they were both very different, which is echoing Carmen's words as well. But I owe so much to the time that I spent with them and the curiosity that they stemmed in the way that, I mean, how could two boys think so differently and see the world so differently compared to me and compared to people I knew? So this was really the reason why I ended up being a researcher and ended up working in autism. So I thank them. So I wanted to begin with a really beautiful video, and apologies if you've seen it, but I've seen it a few times now and I like to watch it again and again.
We are all different, and that's wonderful. Some differences are easy to see. Height, hairstyle, gender, eye color, and so on. Other differences can't be seen. Our favorite foods, fears, or special skills. Interestingly, the way we see the world is also different. For instance, what do you see in this drawing? Most people see a duck, but some of you might have seen a rabbit. Whichever you saw, you are correct. This is just a trick drawing to show you that all brains work differently. The brain is your body's computer. It works differently for all of us and controls how you learn. That's why we are all good at different things. How you feel, which is why we all feel different emotions. And how you communicate. Sometimes the brain is connected in such a way it affects the senses and how we perceive and read situations and interactions. This is known as autism. Many people have autism, so it's likely you already know someone who is autistic. And for this reason, it's useful to know a little bit about autism. The special wiring inside an autistic brain can sometimes make the person good at tasks we may find difficult, such as mathematics, drawing, or music. It can also do the opposite, and activities we find too easy are incredibly difficult to them, such as making friends. The senses constantly send information to your brain about your surroundings and other people. However, when a person's brain and its senses don't communicate well, the brain can become overwhelmed and confused, affecting how they see the world. Picture yourself walking down the street. This is how an autistic brain may experience the same walk. Scary, isn't it? Sadly, in many cases, the person can't say out loud how they feel. So even though there's chaos going on in their heads, they seem okay on the outside, unable to ask for help. We all develop behaviors to help us feel calm in uncomfortable situations. We may look away, hug ourselves, chew our fingernails, fidget, bite our lips, and so on. Equally, autistic people develop behaviors that help them cope with these intense moments. These actions may seem unusual, but they're just their way to feel calm. When they happen, it means they are having a hard time. The kind thing to do is not to give them an even harder time by getting cross, ignoring them, or mocking them. Remember, just because a PlayStation can't read an Xbox game, it doesn't mean it's broken. People with autism need friends who are willing to take the time to know them. With good communication and plenty of patience, everyone would be better off. People with autism are not ill or broken. They simply have a unique view of the world. And with a little support from their friends, they might just be able to share that view with us. Autism can make amazing things happen.
I think that video is beautiful, and it conveys the real need for why we need to understand what makes our brains different. So let's look at some brains closely. So brains come in many different forms. Some are very, very simple and only have, say, 800 neurons. And then as we move up the evolutionary chain, they get more and more and more complex until we get to, this example might be debatable, but 86 billion neurons. At the beginning of our lives, our brains are a little bit like a piece of photographic paper. They are unexposed and white. And it's when we're born that we're exposed to the light and the photographic paper starts to develop patterns and shapes. So at the beginning of our life, we actually have these 86 billion neurons. But what accounts for brain growth? What accounts for all the new skills that we learn from the newborn stage all the way up until 3, 4 years old? It's the connections between these brain cells. And this is really, really critical that the connections are made in the correct way, but there are many different correct ways that a connection can be made. Here is a beautiful walk through a brain. And you're looking at brain cells, but only 10% of them. There are so many brain cells, and the connections are just unbelievably beautiful and unbelievably complex. Now, clearly 10%, this is one example, but here is an example of what two brain cells look like when they connect with each other. And as you see new colours layered on top, these are other connections and support cells. So I'm showing you this progression closer and closer to the point where these two connections happen to convey how complex this is, how wonderful it is that it often works, but how baffling it is for researchers like myself to try and unpick which connections are essential for brain development and for function, and which ones can be tweaked and which ones can be different. So going back to these brain connections, interestingly, they actually prune back after the age of 3. So it's not that we need lots of them, but we just need the correct ones, or enough of the correct ones to be able to function in a way that helps us navigate through life. So what does a neuron look like? A neuron is a brain cell. Here we have a video, and we're going to go deeper into the actual connection between two brain cells. So the little tiny black dots are called receptors, and they receive a message from the neuron upstream. When the neuron upstream becomes excited with electricity, it releases chemicals. So you see the excitation there, the clearing of the area in between, which we call the synapse. And these balls are vesicles, they're bubbles of chemicals that allow one brain cell, one neuron, to contact another one and send a message across. The chemicals are tiny. When they bind like a lock and key, the brain cell below gets the message that it needs to change its electricity and forward that message down. So that's what we're dealing with at the level of a synapse, that connection between two brain cells. So I'm really, really interested in what makes a brain different. And in particular, what kind of connections do we need and which ones can be different and potentially switch but are still functional. So when it comes down to it, what makes a synapse functional? It's actually all down to our genetic code. So our genetic code is responsible for the initial wiring of the brain. It is very malleable after that, but down to the level of genes, we need that gene to tell us what types of building blocks that we need in that synapse. So we can represent DNA like a string of letters, which you can see just here at the end. And a little change in some of the letters can change whether that DNA has a code for a functional building block or for a dysfunctional one or for one that's very different. And I like thinking of this analogy. So the quick brown fox jumps over the lazy dog. If we delete a little bit of this, you still read it in the same way. We might translocate a little bit of a quick brown fox to another bit, and then we get a lazy dog. Or a lazy fox dog, should I say. A substitution is a really small change in DNA, and this results in sometimes a completely different function of that particular building block in our brains. So in this case, we get a quick brown box, which is a really hilarious image, I think. So genetic changes are actually important. It's what gives us edge. It's what helped us evolve. It's what actually makes us different. And in the case of this young individual, it's probably what gives him this unique ability to fly over a city and then draw almost to scale every single building in that city. This is New York. So in some cases, though, genetic changes are not advantageous. So autism runs in families, we know that, but there's no specific genetic cause. We cannot find a single gene. But we can find some very rare cases of a gene that links very, very strongly to autism within one family. So researchers are very interested in taking this small genetic change to understand more about how this genetic change can impact on the connections that are made in the brain. And then hopefully we can transfer the information we get from that research study back to the general population. So this is a picture of a synapse here, and the detail isn't important, but you can see that a lot of the pieces of machinery in this synapse are coloured in red. And all of these red pieces of machinery are implicated in autism. And I'm particularly interested in one called neuroligand, just there on the left-hand side. So the most remarkable thing about this particular genetic change that we're interested in is it's so tiny. So if you have a look up there at the string of letters, our DNA, so can you see that I've just changed a tiny little letter in the code? That's the level of the genetic change that we're interested in. So this genetic change was found in 2 boys from a cohort of people in Sweden. And these 2 boys had autism. And it was the only genetic change that could be found that linked to their autism. So when this change occurs in the DNA does not function in the same way. And it impacts on the way that this neuroligand piece of machinery, which is seen in purple, can connect and function in the synapse. I like to think of neuroligand as a piece of synaptic glue. It keeps the synapse together, but also you can see it's connected to a lot of other pieces of machinery. It's very important for recruiting the things that this synapse needs to work at its optimum. So we can't look at connections in people to the same level as we need to be able to understand this diagram here. We've got magnetic resonance imaging, which gives us a really good idea about the highway connections in our brain. But we can't look at the level of the connections that I showed you in that image before. This is an image that was similar to the neurons I showed you before. This is from the mouse. So we turn to the mouse brain to be able to understand more about how the intricate connections between neurons are formed. And we can stain them with beautiful fluorescent dyes. And the reason why we can stain them is because different neurons have different pieces of machinery on the outside of their cells. So we're able to identify which neurons want to fire more often and which neurons actually don't want to fire with electricity. So what can our mice actually tell us? So we have introduced this tiny little gene change from the 2 boys into the mouse's genetic code. And we've been looking at their behaviour for a long time now. So mice are 85% similar genetically to us. So they pretty much have an equivalent gene for every gene that we have in our genetic code. So we can use them as a way to understand how slight changes can impact on the way that brain connections connect. In addition, they actually exhibit quite a lot of behaviours that are like the social behaviours that we look at in the clinic when psychologists are looking at people who have autism. And my team have been looking at the NL3 mouse, which is the same. So NL3 is my little acronym for the neuroligand gene change that I was introducing before. So I'm just going to play you a couple of things here. So initially, when we looked at our mice, we looked at younger mice. So we put 2 mice together in a cage that had never seen each other. And our NL3 mice were very quick to initiate social contact. And they were very, very likely to chase the other mice that didn't have the gene change. The mice that didn't have the gene change ran away. So this was the first glimmer that we got that maybe they're not interacting in the same way. When we followed them into adulthood, our mice, instead of chasing, they turned to aggression. So they were already showing differences in the way that they interact with other mice. Our NL3 mice also sang a song that was different. And many people haven't actually heard this secret mouse song because it's actually 10 times higher than our brains can perceive. So we use a special microphone similar to the one that you would use to pick up a bat call in the ultrasound. And we can eavesdrop in on our mouse's ultrasonic language. And this is an example of what it sounds like. So this sound recording has been pitch shifted down 10 times and slowed down. It's a very fast language. Each one of these little syllables or calls can occur at 10 to the second. So we do need to slow it down so we're actually able to look at it as scientists. So really fascinatingly, our NL3 mice use different calls. They don't use the same calls as those mice that don't have the gene change. So we're getting some really unique results coming out of this particular observational study that tell us that our mice are not behaving like mice that don't have the gene change. So the brains of our mice are also connected differently. And we know this because we can look at the level of their synapse. They have different levels of machinery in their synapse, so we can see quite a lot of the story coming together here. But my team are very interested in looking at perception. So going back to that little section of video, what is it about perception that is different between our NL3 mice and our mice that don't have that gene change? Could differences in, for example, sensory perception and attention be the reason why they don't interact in the same way? So we know that people with autism display perceptual strengths and weaknesses, and these often are in the visual or the auditory domain. These can manifest as altered attention, and often you will get a diagnosis of autism and ADHD in the same individual. But it's not easy to shift through these studies from the clinic, because what we end up seeing is that some people with autism have perceptual strengths. They're much better at attention tasks, in particular visual search. Some people with autism are actually more distracted. So when you put them through an attention task in the clinic that has a lot of variability and a lot of unpredictable things happening, you increase their sensory load and they cannot perform at the same level as people who are neurotypical. So because there's such noise in the clinic, we wanted to look at attention and perception in our mice to see if we could learn more about why they're interacting differently. But how do we look at perception in mice? We actually want to look at perception in mice in the same way as what we do in the clinic. So this is a video of our mice performing, it's very fast because I've sped it up, but performing a clinically relevant task. So you're looking at a bird's eye view of a mouse in a triangular-shaped box, and it is operating a mouse iPad. It is nose poking a stimulus, and when you see it running to the back of the triangle, it's getting a reward. And we motivate our mice to perform using strawberry milkshake because they really like it. Now, you may be able to work out this task by looking at it multiple times, although potentially you might not be able to. It's actually a really difficult task. What our mouse needs to do is it needs to match a particular stimulus, either the plane, the spider, or the flower, to a particular location. And every time we get two images shown, one is correct and one is incorrect. So this is a clinical task that's used specifically in dementia, and it is really sensitive to early dementia. This is not one that we use quite regularly in our mice with attention, but I'll show you that next. So this is an attention task. So the mouse is wandering around, it's waiting for a stimulus, it got it, and it got its reward. So we train our mice to do this 50 times a day for many months on end. So they become really, really good at this. They are very, very quick to respond, and the stimulus is only shown for 2 seconds. It's just a brief flash of light. So we call this the 5-choice serial reaction task because there are 5 choices. Now, after our mice initiate a task, so they need to nose poke the little area where they get their strawberry milkshake, we give them a delay, a flash of light, and then we ask them to respond. Now, their response can either be correct or incorrect, but they can actually miss the flash of light completely, altogether. And this gives us a measure of how distracted they are. We also want to know what happens when our trained mice that know exactly what to do are pushed. So we change the delay, and we change the brightness of the stimulus, and we also change the stimulus duration. The hardest duration is 0.2 seconds. So this means a 0.2 second flash of light on the monitor. And again, we're very interested in looking at, did they respond? And if they did respond, did they get it correct or not? So I'm going to show you a figure now, and everything in red is a decrease relative to those mice that don't have a gene change. And everything in blue is an increase. So you can see here that trained NL3 mice, don't respond all that often. They actually respond less than the mice that don't have the gene change. So we can interpret this as a distractible phenotype. But then, when they do respond, they're more correct. They're very slow to respond. So when we measure the time it takes for a mouse to make a response once the stimulus flashes on the screen, the NL3 mice are withholding their response, they're thinking. Pink means no change whatsoever. So accuracy is when we're really pushing our animals. So what we're doing here is we're changing that 2-second flash of light to very, very brief flashes of light, all the way down to 0.2 seconds, which is really hard to do. I find it hard to see. And we see the same thing. Our mice are very, very unlikely to respond. And when they do respond, they're actually withholding their response. In this case, we don't see that they're better. But we do get a glimmer that they might be better at the delay version, which is when we change the predictability of the stimulus. So this task here is very simple. Most people here would be able to respond to a flash of light on a screen. So we wanted to do a task that's actually directly applicable in the clinic, and it's this one here called the continuous performance test. So what mice have to do in this one is respond to one image and inhibit their response to another 4. So the mouse here on the left is on vertical bars, and the mouse on the right is on horizontal bars. And I'm just blown away that mice, they're such simple creatures, are able to withhold their response and inhibit the touching of the screen to the distractor stimulus. So this is a task that's done in the clinic, and in particular, it's done over a long period of time. So our mice get to play in these boxes and respond for 100 trials a day, again for a couple of months. So we have a lot of data from one individual animal, but of course we look at many different individual animals to be able to get the collection of data. So once the mouse sees the image, delay, and this is an example of some of the distractor stimuli. So if the mouse is rewarded for the horizontal bars, they're the ones that it needs to inhibit its response to. The stimulus duration is 2 seconds, but we're also very interested in pushing our mice, like we did with the previous task, and changing the stimulus duration to make it shorter. And we're interested in looking at, did they get it correct? Were they accurate? And also their response. So the first graph you can see is whether they got it correct or not. Now, even though there's a small, I'm not sure whether you can see, it says baseline 2 seconds. So the 2 bars on the left-hand side, the black is the mice that don't have the gene change, and the red is the mice that have the gene change. So you can see they actually are better at discriminating the correct image from all the other distractor images. But when we really push them and take it down to 2 seconds, our mice lose this ability. And when you look at their accuracy, their hit rate goes down too. So this is the second graph. And their response is exactly the same as the previous task. So even though they're actually able to accurately pick up the distractor images versus the correct images, they're not responding as many trials. So they're actually missing trials. So this was really amazing that we were able to show the same thing in both of these tasks. One of them was a more simple version, but then the other is directly from the clinic, the same sort of task that a psychologist would use to look at attention in a person. So where are we going now? We've learnt that NL3 mice are better at attention tasks when they're trained. But when things are not predictable and the sensory load goes up, our mice lose this advantage. We still don't know what connections are important for attention, and in particular, we don't know how these intersect with the connections that are important for sensory ability. And we don't know how a very small gene change can impact on these. So these are the things that we're following up next. And in particular, I think that this might help us understand what enables us to be most calm and for people to be able to listen and learn in an environment. And I'll just finish off with this, because I think this is the most
The time to
video that I played before, and that's
With good
understanding how different brains are wired and why some things are easier or difficult is really important. And this is going to actually help neurotypical people to interact with people with autism, because there is definitely a lot that we're missing out on. So last but not least, I'd like to thank a big team of people that work on this work. I showed you a snapshot of the data that we have. There are many, many more things that we're doing around social interaction and also higher order attention. And these are some of my students here, but most of them are listed down on the left. And a special thanks to my mentor, scientific mentor, Professor Anthony Hannon, and all of the people that support us in our animal studies. So we have a lot of staff that help us keep our animals happy. Believe it or not, keeping a group of mice happy costs as much as it does to keep me happy a year. So it's very expensive. And also we must thank Carmen and Quinn for their words, because that's a really generous way to start the talk and really beautifully framed why it is that I want to do the things that I do. So thank you very much, Carmen. And thank you for your attention. And does anyone have any questions about the data, about autism research?